Hi, my name is Jules Stacy, and this is a presentation on neuroevolution of augmenting topologies. Before discussing neuroevolution, I would like to give a short background on neural networks. Neural networks are a form of unsupervised machine learning directly inspired by the human brain. They are composed of interconnected nodes that are arranged in layers. As the data moves from layer to layer, each node transforms the data using a weight. And if the weighted and transformed data is above a threshold, the node fires, sending a signal to the next layer. The first layer in a neural network is always an input layer, the last layer is always an output layer, and the layers between are called hidden layers. These are some of the important hyperparameters for neural networks. During network initialization, weights are randomized. The batch size is the number of observations per step in the model, and the learning rate is the number of steps the model takes before the model weights are updated. Epochs are the number of times the algorithm reads the full data set. The number of layers and nodes present in the data is typically determined through experimentation, and the optimization algorithm options differ greatly from each other, but are based on gradient descent methods. Evolutionary algorithms are a means by which neural network training can be enhanced to create more effective models. For model evolution to happen, a series of possible solutions are first created and stored, which determines their fitness. Each solution is then encoded into chromosomes with strings of ones and zeros. Finally, the solutions are evolved through either mutation or crossover. Mutation occurs when a bit in the chromosome is randomly switched from 1 to 0 or 0 to 1, and the crossover occurs when strings of bits are traded between solutions in such a way that the trades are biased by fitness. This concept will be further explained in a later slide. Regarding neural networks, topology refers to the structure of the neural network, defining how neurons connect to each other. As an example, I will explain a fully connected 3-layer feed-forward network. Fully connected indicates that the nodes in one layer are all connected to nodes in the next layer. 3-layer indicates that there is one input layer, 2 hidden layers, and an output layer, since the input layer is not counted. And feed-forward means that the data moves in one direction through the network. So this raises some questions. What is the best topology? How many nodes and layers should you have? And should all the nodes be connected to each other? And since the number of combinations is prohibitively large, how would someone go about finding an optimized topology? The answer to these is to use evolution to evolve the neural network topology into an optimal state. Where traditional neuroevolution methods use fixed topologies that are fully connected and do not change with a predetermined number of nodes and layers, topology and weight evolving artificial neural networks, or TWIANs for short, evolve both the topology and weights and make changes to the number of nodes and hidden layers, as well as the connectivity. This results in a neural network that is optimized with respect to both topology and node weights. In 2002, Stanley et al. Published Evolving Neural Networks Through Augmenting Topologies, which introduced the concept of neurological topology evolution. They found that evolving both the topologies and connection weights of these networks significantly enhanced their performance. They set out to provide a way for dissimilar topologies to exchange genetic information, to avoid dismissing premature topologies before they were optimized, and to minimize topology complexity without a fitness function. This was achieved by starting with structures that were minimized at initialization and incrementally grown. Much like the difference between a juniper bush and a bonsai tree, this results in less bloat, less pruning at the end, and ultimately an optimized model. As previously mentioned, the first method of topological evolution for neat models is mutation. Mutation can occur in 2 ways. The model can either add a connection, shown in the top half of the figure, or a mutation can add a node, as shown in the bottom half of the figure. When a mutation results in an added connection, the model connects two previously disconnected nodes and adds a random weight to the connection. In the top example, nodes 3 and 5 are connected to each other. Chromosome 7 is then added to the chromosome list above the network diagram, showing the connection from 3 to 5. I use chromosome and gene interchangeably. When a mutation results in an added node, the added node is connected to an input and an output, and the old connection between the nodes is disabled. The input weight to the new node is equal to the old weight value, and the output weight is equal to 1, indicating a passage of all data to the next layer. This output weight is, these input and output weights are then updated on the next step. As shown in the bottom example, node 6 is added, and then a connection is made from node 3 to node 6 and node 6 to node 4, and then the connection from node 3 to node 4 is disabled. The second method of topology evolution for neat models is crossover. In the simplest terms, crossover occurs when two parent networks create a child network through the sharing of genetic information. In the above portion of the figure, parent 1's chromosomes are shaded gray and parent 2's chromosomes are unshaded. Chromosomes 1 through 5 are identical between parents 1 and 2, and the offspring will receive those genes. The offspring will also receive gene 8 from parent 1 and genes 6, 7, 9, and 10 from parent 2. 6 and 7 are considered disjoint genes, as is gene 8. This is because they occur in the middle of the string. Genes 9 and 10 are considered excess because they occur outside of the scope of from 1 to 8. They are in excess of the list of genes. Sharing these disjoint and excess genes results in an offspring network that contains all node connections present in each parent network, which is not fully connected and which will behave in a radically different manner. Additionally, it receives chromosomes that were previously disabled from structural mutation. Finally, the neat method goes further by allowing for speciation of networks. It pairs similar topologies into similar species using historical markings, as well as an excess and disjoint genes that were passed from parent to offspring. Each species has a niche at which it is most fit, and organisms in a given species must all share this niche. The size of each species is capped, which prevents overrepresentation in the total population of organisms. This allows competition within niches while protecting topological innovations by allowing time for each species to optimize before being judged for optimization. Thank you very much for watching. If you have any questions, please feel free to reach out to me. My email is shown.